Hi everyone, I'm Thomas Cavallaro.
I'm Grace Frey.
I'm Hayat Mohammed.
And I'm Brianna Agomessou.
And our virus to study was HIV. To first understand HIV, you have to look at and understand the basics. HIV stands for Human Immunodeficiency Virus. It attacks CD4 cells, which are helper T cells. They are the primary defense of the human immune system, and their function is to ironic infections. HIV virus starts off with binding, then fusion, reverse transcription, integration, replication, assembly, and budding. There is no cure for this virus, however, treatments are available. The most common form of treatment is ART, and it can stop the spread of the virus. This gives people the chance to live long lives. This is a global epidemic, as you can see, so it begs the question, what makes this virus so dangerous? To answer this, we have to dive into the makeup of the virus.
So, at the basic level, HIV is two identical single-stranded RNA molecules that are each 9,200 to 9,800 nucleotide bases long. It starts off with the GAG and the POL genes on the left, and those code for different enzymes that the virus uses to attack the cells. There's also at least 9 different proteins in each strand of the RNA, and these fall into 3 different categories. The structural proteins build the inner membrane and the capsid of the virus and are essential components of the structure, allowing it to move between cells and infect different CD4 cells. The regulatory proteins are necessary and responsible for the production and replication of new viral HIV RNA and help in splicing and binding. And then the accessory proteins are not required in every strand, but influence the overall rate of the production of the virus and can help it attack faster.
The reason why this virus is so deadly is because of its life cycle. HIV has a life cycle like all other viruses, and the main goal is to attach to a host, CD4 cell, and reproduce. It is a 7-step cycle. It starts off with binding. The HIV virus attaches itself to the CD4 antigen receptor on the surface of the cell using its own surface glycoproteins. Next is fusion. The HIV envelope and the CD4 cell membrane fuse together, and the HIV viral genetic information and all of its enzymes enter the cell. The third step is reverse transcription. Now that it's in the cell, it uses its enzyme reverse transcriptase to copy its viral HIV single-stranded RNA into HIV double-stranded circular DNA. This now allows the virus to enter the cell's nucleus. The next step is integration. Now in the nucleus, the HIV DNA releases an enzyme integrase, which allows itself to add itself to the host cell's DNA. It's now one very long strand of DNA and is now called proviral DNA. The next step is replication. The HIV virus uses the CD4 cell's machinery, RNA polymerase, to make long chains of HIV proteins. It can replicate into messenger RNA and viral genome RNA. The next step is assembly. The HIV proteins and RNA move to the surface of the cell and form together to an immature HIV cell, which is non-infectious. The final step is called budding. Finally, it releases itself from the cell and the enzyme protease is released. This enzyme breaks up the long protein chains and cuts the HIV protein chains into its mature infectious vials. These contain all 3 necessary enzymes and viral genome RNA and move on to other CD4 cells to repeat the process. This process occurs very rapidly and can create problems quickly because it attacks the immune system directly. The helper T cells are attacked, a very key cell in the immune system response. This virus is also very dangerous because of how the virus is made up. The RNA and readily available enzymes make it easy for the virus to attack and spread rapidly.
For the mode of infection of HIV, HIV is found in the bodily fluids of someone living with the virus. These fluids include blood, semen, preseminal fluid, rectal or anal mucus, vaginal fluids, and breast milk. In order for HIV to be transmitted from one person to another, these bodily fluids of an infected person must enter the blood of another person. Transmission may occur through a mucous membrane, through cuts or sores in the mouth, or through sharing injecting equipment like needles. A common misconception is that bodily fluids like saliva, sweat, and urine are able to transmit HIV from one person to another. But there's actually not enough of the HIV virus in these bodily fluids of an infected person. Treatment can be taken to lower the amount of the virus within an HIV-positive individual, making the virus undetectable. This means that the virus cannot be detected by a normal blood test. but a person living with a detectable load of HIV can transmit the virus to others regardless of the presence of symptoms. HIV is most easily transmitted within the first few weeks of infection because many are unaware of their status. The nature of the effect on the body is that there are 3 stages of HIV. Acute HIV is when flu-like symptoms occur days, weeks, or months after contracting HIV. Chronic HIV, or the asymptomatic stage, can last several years. The last and most aggressive stage of HIV is AIDS, which occurs when CD4 cells count fall below 200 cells per cubic millimeter. This makes a person at risk for future infections. For acute HIV, it is an infection that takes place immediately after contracting HIV. Symptoms can take days, weeks, or months to show up after the transmission of the virus, leading to the virus multiplying rapidly. The initial stage of HIV can result in flu-like symptoms, including fever, headache, rash, swollen lymph nodes, fatigue, and muscle pain. These symptoms are due to the increased copies of HIV and extensive infection in the body. When this happens, the amount of CD4 cells begin to fall quickly. Once the immune system kicks in, CD4 levels start to rise again, but they may never return to their pre-HIV height. For chronic HIV, also known as the latent or asymptomatic stage, it has various effects on the body. During the chronic stage, a person's symptoms will decrease compared to the acute phase due to the virus not multiplying as quickly. During this stage, an individual is still able to transmit HIV if it's left untreated. Without treatment, the chronic HIV stage can last for years before advancing to AIDS. AIDS is the late stage of HIV infection that occurs when the body's immune system is badly damaged because of the virus. In normal adults, the CD4 count ranges from 500 to 1600 cells per cubic millimeter of blood. A person is diagnosed as having AIDS when they have a CD4 count of fewer than 200 cells per cubic millimeter. Individuals with AIDS are vulnerable to future infections. Common infections include tuberculosis, pneumonia, and etc. Without HIV medicine, people with AIDS usually survive about 3 years, but medicine can still help people out at this stage.
So overall, HIV has a lot of connections to the biology learning outcomes, but the 3 biggest one it hits is describe characteristics of viruses and bacteria, describe the nature and function of enzymes, and describe major mechanisms used to control their activity, and describe the nature and expression of heritable information at the molecular level, including DNA replication, DNA repair, transcription, and protein synthesis. So first, this pathogen is an excellent example of explaining how a virus enters a cell and changes its DNA to replicate and attack the human immune system. It contains the basic units of a virus, such as its capsule with all of its enzymes and binding proteins that display how viruses work at the cellular level. Also, this pathogen uses reverse transcriptase to change the transcription in a healthy functioning CD4 cell. These enzymes synthesize a DNA version of RNA genes. This modification of the central dogma using an enzyme creates this reverse information flow, allowing the virus to function. And finally, HIV targets the transcription of RNA and translation into proteins in normal CD4 cells. This also reflects the disruption of the natural functioning of the central dogma and displays the way replication can be altered. Protein synthesis, the final step in this virus transmission, is essential to spreading the virus within the immune system.
So current research in the field. Current HIV antiretroviral medications can only help to stop replication and transference, but it does not actually cure the infection. But if patients that are undergoing treatment have the treatments interrupted, it can cause for health issues and flare-ups in the patients. This causes patients to have to continue treatments forever because interruptions can be detrimental to their health and cause other problems in their life. Scientists are trying to work towards a cure to inactivate the HIV DNA by looking at new therapeutic techniques. The CRISPR-Cas9 genome can be altered to help strike against HIV DNA. In the figure, the Cas9 gRNA is attracted to the HIV DNA and it attacks it. It is also depicted that mutations that are dangerous to HIV will lead to a cure, but it is also noted that some mutations are not dangerous, which means they won't fight against the DNA. It would only cause for a viral escape. Other research is still happening in this field, and they're trying to still find a cure for HIV, but this is just a very prominent thing that's happening right now. So thank you for listening to our presentation, and if you have any other questions, let us know. And here are our references.